Hello and good evening. My name is Axel Glasmacher, and I have the pleasure to welcome you to tonight's CDDF's webinar about matching endpoints and objectives in clinical trials, reflections on disconnects and opportunities. Our speaker will be Armin Schüler, and we will present him in more detail in just a moment. The CDDF is the Cancer Drug Development Forum. It is an international non-profit organization registered now in Belgium that provides a neutral platform to stimulate interactions between all stakeholders involved in cancer drug development. That includes regulatory agencies, HTA bodies, academics, patients, and others. So we at the CDF, we bring together leading voices from academia, pharmaceutical industries, regulatory authorities to improve cancer treatment. Please visit our website at cddf.org to get to know more about us and our upcoming programs. We have a few technical aspects. So first, we've put all of you on mute to preserve good audio quality. There is a chat function in Zoom, and you can use it to report any technical problems to the CDDF. Our staff is there and will reply to you, and so we can work on any issues that could occur. You will also see a Q&A symbol on your Zoom bar, and that, please use that one, not the chat, to ask questions. You can ask questions at any time. We will, however, only address them at the end of the meeting. But if during the talk you have an interesting idea of a question, please send it immediately so your mind is free for the talk and for the next questions. You can either watch this in a limited screen or in a full screen mode, and double-clicking on the Zoom screen would allow you that. This meeting actually was conceived as a preparatory webinar for our multi-stakeholder workshop that will go ahead in April. It will be a virtual meeting due to the COVID pandemic, but we will have additional workshops there on endpoints in cancer drug development, the controversy around overall survival, expedited approval pathways, and patient-reported outcomes. And obviously, any learnings we take away from this webinar will also help us to tailor the workshop more to your demand. Please go to our website. You can get all the information you need there, and you can also find ways to stay updated on this development. The moderator of today's meeting is Robert Hemmings, and we are very happy to have him. Robert is a partner at Consilium, a consultancy partnership between him and Thomas Samuelsson. Both are long-standing contributors to the EU regulatory network. He is a statistician by background. He has worked at AstraZeneca and the MHRA in the UK for 19 years. He was for 11 years at the CHMP and has served as rapporteur on multiple products and engaged in intense scientific and policy aspects of the committee. He chaired the CHMP Scientific Advice Working Party for 8 years and worked in EMA expert groups on biostatistics, modeling, and simulation. So I don't think we could have a better moderator for today's topic. And I'll turn over to you, Rob, and to present our speaker and to moderate the rest of the session. Thank you very much.
Thanks, Axel, and hopefully you can hear me well. It's a pleasure to support the meeting today, and I thank you and the CDDF team for hosting today's session on a topic that I think will be an evolution of clinical drug development in some areas, and will actually be a revolution in other areas. Back in about 2015, I was privileged to be working as part of the EU regulatory network, as you've just heard, and we initiated an update to the ICH E9 document, Statistical Principles for Clinical Trials, because of a concerning lack of clarity in clinical trial objectives and a perceived misalignment between the objectives that were relevant to regulatory decision making and the analyses that were being conducted by sponsors. The solution to which was a more precise description for the target of estimation for a particular trial objective, referred to as an estimand. What is to be estimated? And an important principle that we should talk about what is to be estimated before we talk about how to estimate, so the statistical methods. This is the agenda for today, and as you've heard, we're fortunate to hear a presentation from Armin Schuler. Armin will be known to many of you, I'm sure, a distinguished career in the pharmaceutical industry and currently head of global biostatistics oncology at Merck. And per Axel's introduction, please do feel free to put your questions in the Q&A section of the webinar tool, and we'll monitor those and we'll grill Armin at the end of his presentation. So without further ado, Armin, we give the floor to you.
Okay, welcome to the CDDF webinar on matching endpoints and objectives in clinical trials, also from my side. The new ICH E9R1 guidance is a trigger to reflect on the link between objectives of a trial and, let me rephrase, the research question of a trial and the endpoint. Today, we are often focused very much on the endpoint. At the end of the trial, for example, in a press release, it's often stated the trial met or did not met its endpoint. Nothing is said about the research question. In my view, the question is not to meet and greet the endpoint. It's a question whether the trial was able to answer the research question. I will come later back to this, why I think it's not the same. You see on the right that 5% of the drugs which go to phase 1 will lead to an approval. This is not an issue per se. That's why we run clinical trials, to make sure that non-effective drugs don't make it to the market. It shows us at the end that clinical studies are still necessary. It's a failure rate of phase 3 trials which are concerning. You see here, only 40% of the trials are positive, met their objectives, and it's independent across cancer types. Okay, pancreatic cancer is a little bit lower, but we all know the challenges of pancreatic cancer. Or for example, you might remember the failure of the ECHO 301 trial in advanced melanoma, when the phase 1-2 response rates over 50% were seen, but in the phase 3 study, the response rates were only 1/3, and the results were similar across the arms when combining epacoudostat with pembro. To be fair, based on the results of the 202 study, I would have also recommended to go into phase 3. And I remember a discussion during an IDO advisory meeting just after the results were released, where the discussion was a kind of, oops, we still have to run randomized trials. And it was at this time, we are in the pembro, who needs randomized trials? So, trials often fail to show what we are expecting them to show. Potential reasons are impact of the study population. Different study populations included in the phase 2 compared to the phase 3. This could be because change of study population by in-exclusion criteria, obvious, change of the landscape by approved drugs in the subpopulation, leading that in the complement, the non-approved part, more people go into the trial, or competing trials with overlapping populations. Or change of centers. For example, the phase 2 trial had a focus on academic centers. There, either patients could be more healthy because they have to be able to travel, or they might be even more ill because, let me say, non-severe patients were treated at the local hospitals where the severe cases were moved to the academic center, or difference in smoking habits, East European and West European, leading to different prevalences on NSCLC cancer subtypes, adeno versus squamous. Impact of outcome measurements selected for the individual trials across phases. What we see in objective response might not translate into an effect in OS. Using an independent read might also have a moderate impact on the individual cases. I will come back to this later. Or coming back to the echo example, for single-arm trials in drug combinations, the benefit of the partners of the combination might turn out to be different than expected. The contribution of the drug is clear if you see responses on monotherapy in late-line patients without any treatment options. Those cancers are not shrinking by chance. In case of combination with a potent partner, it's not that straightforward. Or I have to highlight this as a statistician. For example, if you run multiple single-arm trials and run a pick the winner strategy, you might run into a chance finding. Or let me rephrase, you are rather not going into the next phase if you have an underestimated effect observed. And 5, you will know yourself a lot of other reasons. And this is just a subset of reasons. So, and now, what's to do? Let's step a little bit back and reflect on what we are doing. In 1962, John Tukey, some call him the founder of exploratory data analysis, for example, box plots were introduced by him, stated in an article, at that time they were 62 pages long, easy to read, far better an approximate answer to the right question, which is often vague, than an exact answer to the wrong question, which can be always made precise. It's about the research question objective and not about the endpoint. It's the right answer and not the exact answer. In our days, we are sometimes focused too much on an exact answer that the research question sometimes get out of sight. And to be fair, do you have always in your trial a detailed discussion on the statistical methods with the statistician? And it starts with simple assumptions. For example, Kaplan-Meier assumes that censoring is at random. This cannot be taken for granted. So, are we asking the right question? Just a simple example for the endpoint overall survival. So question number 1 could be to demonstrate improvement in overall survival with treatment A compared to treatment B, followed by subsequent therapy. With other words, what is the drug adding to the existing landscape? That's what we normally estimate today, and we are what we normally estimate today when we are analyzing OS by means of the ITT principle. Another question could be to demonstrate improvement in OS with treatment A compared to treatment B if there would be no further line treatment existing. With other words, what is the benefit of A compared to B if not diluted by subsequent therapy? This would be the answer to the question whether the new drug is effective, but it doesn't tell us something about effectiveness. It's at the end an hypothetical question, as in real life, subsequent therapy is always available and will be used. Payers and patients normally are interested in this answer to the question. So the question, what adds the drug to the existing landscape? When we are looking at PFS, it's rather a type 2 question. So, as said, it's about the research question and not the endpoint. Thus, the approach should be how to deal with it. What question are we really interested to answer? This leads to the specific and detailed trial objective. This should lead us to the peacock statement. That means the specification of patients, population, or problem, intervention, comparison, control, or comparator. So it means treatment and outcome. But that it's not the end. Peacock is just a step into the evolution of Esterman's. It's the underlying intention of the talk to introduce Esterman's as introduced by R1. So we are now going to the Esterman's. And these guidelines has been, it's 1 year already old, so you might have heard already something about it. But I will start from the beginning, and don't be afraid. I'm not going too technical. So high level, what is an estimate? So this is direct wording from the guidance text. An estimate is a precise description of the treatment effect reflecting the clinical question posed by the trial objective. It summarizes at a population level what the outcome would be in the same patients under different treatment conditions being compared. An estimator is a method of analysis to compute an estimate of the estimate using clinical trial data. And an estimate, a numerical value computed by an estimator. So, confused? Let's try it simpler. The trial objective or clinical research question and the estimate are the what, while the estimator and the estimate are the how. Let's go a little bit into details. So what's an estimate? It's a target of estimation to address the scientific question of interest estimated as described by 5 attributes. It's population, treatment, variable or endpoint, same meaning in this context, population summary, and last but not least, intercurrent event. Intercurrent events are events occurring after treatment initiation that affect either the interpretation or the existence of the measurements associated with the clinical question of interest. For example, if PFS is an endpoint of interest and patients get next line treatment before progression, this is an intercurrent event. Just because I built the bridge to the PICO statement, it's at the end, the PICO statement with introducing 2 new elements, the summary measure, for example, hazard ratio, and as said, the intercurrent events. So coming back to my overall survival example, classical intercurrent events are the application of subsequent therapy after the randomization, which affects the treatment in an unknown proportion. We don't know what the effect is of the following drug, or treatment discontinuation. So stop of treatment hinders the drug to show its full potential. I will use 2 examples to illustrate. One is the Galerius trial, which compares TNZ versus Beva plus irinotecan. And you see here 2/3 in both arms have been crossed over. It's a notable proportion, so that might influence the overall survival event. But at least here it's balanced. Or a second example, the Javelin Lung 200 trial, where 26% of the patients in the docetaxel arm and 6% in the avelumab arm received subsequent therapy with immune checkpoint inhibitors. Note, the assignment of subsequent therapy is not at random. The treating, you all know that, the treating physician is not throwing a coin. The decision is made on a patient individual basis. And the decision is not always visible from the patient characteristics. So when I'm analyzing such a trial, I have a problem. But I've sometimes seen patterns where shorter PFS time leads to a certain behavior compared to longer PFS time, but it's only a pattern and it's not always clear. So it's a challenge for the analysis. Quick illustration on what this would mean when you would have, for example, progression-free survival, and you want to observe, you have 2 treatments for patient, very small trial. You start the treatment, you have new anti-cancer therapy, intercurrent event, but at the end you are still able to observe the progression. Not to compare with the missing value, the patient withdraw, and you don't know what has happened, or the patient is dying, then you also have an event, or like here, nothing happens, and you can observe the PD as you would like to observe. To see, missing is a different problem. So devil is in the detail. Let's have another look at intercurrent events from a missing lens. High-level summary of this slide is missing data is to be avoided, while intercurrent events are integrated part of clinical practice. For example, when scans are missing or patients get lost, you want to avoid that. While start of subsequent therapy after PD is integrated part of clinical practice, it is a disturbance, clear, but it is as it is. And I gave here, also for the later reasons, some examples, like here, study withdrawal, as I said, or discontinuation of randomized treatment due to an adverse event. So as I said, missing to be avoided, intercurrent events reflect clinical practice. Just to motivate the other 5 attributes, a quick look at population. So I assume the relevance is clear to you. Often in the analysis, then implicit assumptions are made. It's, for example, I often see a statement like pros will be done on the pro analysis set. These are those with at least one post-baseline assessment. And at the end, it's a completer analysis. And there's a hidden assumption that those patients who have filled out a form behave similar like those who didn't fill out a form. And this is not really true. And this is, let me remind you on that, one of the reasons of the estimates framework is to get some hidden things out of the stats section, because who reads the stats section? So let's make it more transparent and visible. The centers I have already reflected on. Or another thing, endpoint is important. You know how challenging, for example, it's to bridge between endpoints. For example, here you see, this is the ratio in the randomized trial of response rates. You see here a ratio from 0.6 to 0.9. On the OS scale, you observe hazard ratios of 0.9 to 0.9 with the same hazard rate, with the ratio of response rates. Or here, it's nearly no difference in response, and you observe something between 0.7 and 1. Or PFS, you see here, these I have highlighted PF trials with some PFS around 0.9, and you see clusters observing around 0.7 or around 0.9. So a lot of challenges, and that's why we have to discuss this in great detail as a team. So coming back to estimates and my starting point, are we asking the right question? I took here an example from renal cell carcinoma where I can have a deeper look. I'm following a paper just published in October. It's comparing 3 studies with renal cell carcinoma through the estimate lens. Here, the scientific question is, does a new treatment prolong patients' DFS time? And it's by intention an example to really go a little bit into the detail. You see here the 3 studies. They are quite comparable, except the treatment, that's clear. But in principle, they have the same research objectives, as I said before, prolongation of DFS time. So let's link the intercurrent event subsequent therapy with the potential clinical questions. I have put here as a reminder these 5 attributes which are important, and I will later go a little bit through the differences in the trials on these elements, which also motivates a little bit why the authors of the ICH guideline have chosen those 5. So, and I will give you here 3 examples which somehow reflect the 3 main estimates introduced by R1. So the first clinical question would be, does the drug delay recurrence if no patients had received subsequent therapy? Classical way to analyze this is to start at a start of subsequent therapy. Similar to the OS example earlier, it is a hypothetical estimate because it envisages the scenario in which the intercurrent event would not occur. We just ignore it. Not ignore it, sorry. We just think it would not be there. Second question, does a drug delay recurrence with or without subsequent therapy? There we use all events even after start of subsequent therapy. That would be a treatment policy estimate, meaning the occurrence of the intercurrent event is considered irrelevant. We just ignore it, we take everything before or after. And for completeness, does the drug delay recurrence or start of subsequent therapy? So one of those. The analysis would be that start of subsequent therapy is an event itself, like recurrence or death, and this is a composite strategy. That means the intercurrent event is considered itself to be informative. So we take this as a failure. In summary, on how we handle the intercurrent event, subsequent therapy, we answer a different clinical question. Sometimes I call this the FDA approach, which is quite common, while the treatment policy estimate follows the EMA guidance on PFS. However, if applied, it might be not the pure implementation. Think you see this in a protocol, you think we take all events, but the protocol is not asking for scans after start of subsequent therapy. If there are no scans, you can't use them. Even if it looks like you would use treatment policy, it's a fake treatment policy. So you have to be very careful, or if you really want to implement something like this, you have then to follow up the patients even after start of anti-cancer therapy in a rigorous way. As I said, I wanted to have a look at all attributes of the estimates. Let's go to the other elements of the estimate framework. Population. It's on the first glance, same population. If you dig deeper, you see here I highlighted the difference. They are not exactly the same. Same is with the endpoint. It's recurrent second primary cancer for 2 trials, but for one, it's local recurrence metastasis. And that's why I also highlighted the composite strategy. In 2 trials, second primary as a part of the endpoint, it is composite, where in the other trial it's ignored, so following the treatment policy. And those different handling have an impact on the event estimate, so the hazard ratio. What else is different in these 3 trials? You see here scan date in case of equivocal findings, either latest, earliest. The assessment schedule is different. And you have here the censoring of missing assessments is different. And the timing of the scan, coming back to the assessment, and how to handle, how to deal with missing scan can sometimes have a notable impact on the effect size. At current, we are doing in nearly all trials a simple approach. We assign the PD time to the time of the assessment. So if the assessment schedules are different, especially across treatments or across trials, when you compare across trials, you might get different results. So the windows in principle have an impact, especially if they, At the same, how to deal with missing my mistake. Yeah, here I am. At it's also important to look at how we deal with missing assessment. For example, take an example. You would only you take all patients into account. So you take all assessments into account. You have, oh no, sorry. If you take all assessments into account and you have no missing value, then you assess PFS in the pure sense. However, assume you have a lot of missing values, and would then you would, instead of the progression, you would observe the death. So your PFS time is rather OS. So if you have a lot of missings and you take all assessments into account, even after missings, your PFS time is the OS time. And I've seen a trial where the effect was flipped depending on these criterion on missing values. So, in summary, estimates offer a structured framework which promotes discussion and increased transparency. It's like a recipe to cook a soup. It clearly describes the ingredients and the way to combine them so that you can compare across soups, and it's irrespectively who runs it. With other words, better alignment between the clinical question and the associated analysis will lead to a more systematic approach to design clinical trials. A better ability to answer the clinical question will ultimately lead to a higher probability of success. Take-home message. Motivation is the guideline itself. You see here a screenshot. What is an estimate? A precise description of the treatment effect, reflecting the clinical question posed by the trial objective, and is described by 5 attributes. Why? Provides a structured framework to strengthen the dialogues between disciplines to improve precision in describing a treatment effect of interest. So as I said earlier, bringing the stats methodology out of the dark of section 9 of the protocol. And when? Always. Okay, required is only for primary and key secondary endpoints in studies with registration and intention, but I would apply it always. Thank you.
Thanks very much, Armin. And you joined us on the video. Excellent. A reminder to everyone listening in to put questions in the Q&A function of the Zoom platform. Just to start with a comment from my side, Armin, to which I'll ask you to react. I think the link into the PICO framework is good, but also is risky. So PICO is a nice structure to define and describe an experiment. And as you nicely described, the population, the treatment conditions, and the outcomes or endpoints or variables are attributes that feed into the estimand, the target of estimation. They help us to define what is to be estimated. But what we absolutely don't want is for the estimand to become a tick box exercise for sponsors and those involved in the design and conduct of trials to think, well, I already know the population, I already know the treatment conditions, I already know the variables, so those parts are done. All I have to think about is this other slightly abstract concept of intercurrent events. And actually that would be, well, both unfortunate, but also not using the Esterman framework to its full potential. Addressing intercurrent events and framing the scientific question in respect of intercurrent events, as you nicely showed in the presentation, might mean that we modify our definition of the treatment conditions or the population that's of interest or the variable that's of interest. That was your composite example. So it's not, this isn't a bolt-on to PICO. This is a sort of fundamental rethink of what do we want to estimate in our trial that happens to involve some of the same ingredients in this soup. I think, as well as the fact that this is now a finalized regulatory guidance, part of the motivation, and at least what I hear from colleagues who are sort of more familiar with implementing the framework, is that this has led to better discussions within clinical teams. As you say, we don't sweep certain issues under the carpet. We don't hide certain problems in the statistical section of our protocols and analysis plans under the heading of assumptions. We shine a light, and that enables better discussions between clinicians and statisticians within the sponsors' clinical teams. And that's also part of the motivation for engaging in this, I think. Also, the fact that people recognize that different questions can be posed. We have now a language to talk about different trial objectives and different types of research questions. You nicely gave some time to event examples around PFS and overall survival. Do we want to know a property of the individual product, a treatment effect of the individual product, or do we want to know the impact of that product when it's put into a broader treatment pathway? There's other types of data, of course, generated in oncology, hematology trials. Safety data, of course. Do we want to analyze a particular toxicity, a particular adverse event based on an ITT principle of all patients with full follow-up? Or are we interested in understanding the treatment effects in patients who are actually dosed for a period of time? Or quality of life data, PRO data, whether they're being used to try and show a symptomatic benefit or to demonstrate the absence of a detrimental effect of a particular toxicity. Again, is it an ITT principle treatment policy type question of all patients with full follow-up of all patients, or is it a question of effects in patients who are able to tolerate and remain on treatment? And of course, an important feature of the guidance document is to avoid naive comparisons of tolerators on drug versus tolerators of control, which would be confounded, of course. So I gave a long-winded commentary to give some time for people to ask questions, Armin. But perhaps you can give some reflections on the comments I've made, and whether there's any points there that you would take issue with or want to build on, and then we'll turn to some questions from the audience.
I want to have few to add to this. I in principle agree to the PICO statement. That was for me, let me say, a kind of driver to introduce a little bit. It's not something totally new to have a structured framework. Some structure has been also implemented in the past, and to highlight what is really the key elements, let me say it like this. In principle, it's, and I'm also in line with you, we have to be careful that this is not a tick box. And when you write a protocol and an estimate setting, it's also important to maybe give the rationale why you choose a certain estimate. So that's clear why you're doing it. Or, for example, I had this example with overall survival with a hypothetical estimate. For example, maybe in your trial, because of the crossover, your follow-up treatment is not reflecting clinical practice. So then the estimating, let me say, this option one, also taking subsequent treatment into account, is not clinical practice. So it's not a good estimation of what you want to know. What is the add-on effect to clinical practice? And then you might use a different estimate. So it depends, let me say. But in principle, otherwise, I'm totally in line with you.
So there's a first question appeared around endpoint definitions, I understand, and the importance of looking at the results of trials according to different definitions that are used. For example, when we assess response, we might have RECIST or mRECIST or iRECIST. And how does the Esterman framework take this into account? And Armin, perhaps you can comment on the number of different Estermans that a trial might include, and in particular where we have different methods of assessment. Are these now different Estermans?
In principle, yes. If you are answering a different research question or an associated research question. Because look, for example, you look at immune-related resist or resist itself, it's a slightly different definition. You have this pseudo-progression issue which drives to immune-related resist. So you are, let me say, the classical resist question would be, I'm looking under the first growing of the tumor, where the immune resist say, I'm not interested in the first grow of the tumor. I'm interested, let me say, as a increase of tumor load, irrespectively of you have an early pseudo increase, and also the tumor load itself as a total is taken into account. So it's nearly the same, but it's slightly different. And that's why it's called in the Esterman framework a supplementary estimate. Not to compare, for example, if you have an independent read and an investigator read. There, they have the same definition, it's the same receipt, it's the same soup they want to cook, it's the same question, it's just two different people who are measuring it. And think about when you see a difference, for example, in immune-related response criteria, in resist criteria, for example, the other one would give in the trial hazard ratio of 0.7, the other of 0.65, then you immediately are able to make an interpretation, and you would, when you have a patient, you can tell something, okay, here the effect is even more pronounced when we go through this making people afraid period of pseudo-progression, that you see all the tumors growing, but there's hope that's going back again smaller. Where when you have resist assessed by the investigator or the independent read, there is not a different efficacy of the drug. It's not, okay, the drug is, investigator read is 0.7 hazard ratio, in independent read is 0.8. It's only working when the investigator is looking at this. Man, it's a little bit joking with it, but it shows you there, different readers, it's just quality. But immune-related response, or I resist and resist, it's a slightly different question. I hope this answered your question.
No, I think so. And then sort of expanding on that, so beyond different ways to define a particular endpoint or measure a particular response to treatment, we may have many different objectives, some related to efficacy or effectiveness, some related to safety, some related to quality of life. Different types of variables that we'd be working with, but also fundamentally different aspects of a sort of treatment or target product profile. Armin, it's not essential to ask the same type of research question in each of those different domains. We may wish to phrase our research question differently for questions about efficacy than we do about safety, than we do about patient-reported outcomes, for example. Do you care to add a comment on the sort of flexibility of the framework, if you like, to work across different trial objectives?
Iman, the advantage, as I said in the speech, of the framework is that to make it more transparent what you are doing. For example, you made the example with safety. If take an overall survival endpoint trial, and now you look at this treatment policy estimate, you look overall survival until the patients die. So you look at the full treatment regimen. When you look at safety, normally you're only looking at the safety under treatment. And even worse, you would have a drug like a checkpoint inhibitor, which is given until progression, compared to a drug which is only given 6 cycles. And often you look only at the time under treatment, you count how many AEs you have, and then you are ready. But it's what are you at the end then again trying to estimate? the safety of the whole regimen. For example, it could be that your drug is very safe, but the next line is chemotherapy and you have more side effects. We ignore that at the moment. Or are you interested, let me say, in incidence proportions or incidence rates? So an adverse itself or the risk to get a certain event in the time period. So the Esterman framework gives you or forces you to be more thoughtful on what you are giving with your number, what you are estimating. So what is, for example, and that's the advantage. Having different populations or different questions, it's totally fine. It's just you have to be honest to yourself. So, and coming back to when you are giving benefit risk, often in overall survival trial, you have the benefit over the whole life of the patients compared to the risk only under the first treatment. Is that a good comparison? You have to think about yourself, but at least it make it visible.
So there can be, and perhaps even should be, a different type of research question across different domains and different aspects of the trial. As you say, the choice we make for an overall survival type endpoint, which may be a treatment policy, what is the overall effect of drug A plus all subsequent therapies versus drug B plus all subsequent therapies. And we may want to do something different in respect of safety. We may want to do something different in respect of PROs. It's about defining.
I didn't comment on PROs.
No, but it's about defining the. I still took your answer to agree that it's about defining and specifying the research question in a precise way that's fit for purpose for the for each aspect of the trial. There's an interesting question around the Esterman's framework outside randomized controlled trials. So the benefit of randomization is hopefully clear to everybody, that at least at the start of the trial, it allows us to be reasonably sure that we're comparing apples with apples, and then these intercurrent events such as discontinuation of treatment or treatment switching or addition of new medications or whatever it might be can happen differentially in the different randomized groups, and that's a reason to be very clear on our research question on our estimate. But not all trials are randomized controlled trials, and some questions are answered through indirect comparisons based on single-arm trials, and other questions are addressed through meta-analyses, pooled analyses of trials. And the question here is, does the Esterman framework make meta-analysis more difficult because of the diversity of Estermans across trials that are essentially asking the same question? And I think that's interesting from a couple of angles. Armin, please comment.
Yeah, I would say the opposite. It makes it easier because it's more transparent. At the moment, if you don't have this information, this detailed information, you make a naive comparison. And take this example I gave. If the endpoint is defined slightly different, or normally it is in publication, or the population is slightly different, and the difference is caused by a prognostic factor, then you can't compare it. And the Esterman framework, if followed by all, makes the differences across the trials visible, and then you can think about what would be the impact of these differences on the comparability. So the data is the same like before the Esterman frameworks. It gives you more opportunity to compare. And this is the same when you compare, which is a part of a meta-analysis, indirect comparison of single-arm trials. If you have take a very easy example, you have scans every assessment is in this kind of part of the population level summary. You have scans every 6 weeks and you have scans every 8 weeks. And then it's a very severe disease. And in one trial, you have median PFS of 6 weeks. And in the other trial, you have median PFS of 8 weeks. And then you say, wow, it's 1/3 better than your drug. But it's just because of the different assessment schedule. And you can only see this if you have all this information.
So Armin, we could actually answer yes. The Esterman framework will make it more difficult, but that could be entirely appropriate because it's just shining a light on a flawed comparison. And as you say, if one trial has estimated the treatment effect under a treatment policy strategy, including the influence of subsequent treatments, for example, and another trial has estimated an effect under a hypothetical strategy as if other treatments were not used, it's risky, it's flawed to combine those estimates in a meta-analysis. And having this, as you say, transparent understanding of what each trial has done shines a light on that and helps us to understand whether we're doing something sensible or not. And it is tempting to say that there's nothing new in this Esterman's framework from one perspective. It is a case of sweeping problems out from under the carpet where they've been pushed before.
Yeah, that's an important point. So I have to rephrase my answer. Yes, it makes your life more difficult, but before it was also difficult. You just didn't observe it. That's true.
So sort of big picture, a big picture question. Is there yet experience across industry in terms of regulatory interactions or interactions with HTAs and other stakeholders? Do we yet have any insights into, firstly, the extent to which each stakeholder group is picking up the framework and using it? And secondly, whether the different stakeholders are starting to show different preferences for different strategies, so expressing an interest in different research questions.
It's a tricky answer. Let me say, my feeling or what I hear and what I partly see is also the agencies. It's a learning exercise also at the agency side. And Rob might even better comment on this than myself. Also there, it has to go out of the stat section to the, let me say, medics community. There might be also a difference between different agencies because take the FDA. The FDA has a big stats department, so they can also within the department train their medical colleagues / go into more difficult estimates. I haven't said anything about principle Stratum, which is also introduced by the guideline. So what see is a mixture. I recently got a question from an agency where I said, to be a little bit frank, oops, I think they haven't looked into the estimates guidance. So it's very diverse. You get, to a certain extent, very specific questions. And so it's an involving exercise, so a learning exercise, at least in my view.
But as you said in the talk, it is at least perfectly possible that the question a regulator may differ from the question a HTA may have, which Correct. May differ from the question a prescriber has, and may differ again from the question that an individual patient has. And the framework does allow us to specify each of these different questions if we've understood them, and to make statistical analyses that are aligned to each different question. I should have put a question mark on the end of that to you, Armin.
I can only repeat what I said, that the advantage that you come from the question and then check what the analysis is compared to let the statistician write a methodology part in your SAP, and then let's see whether, and don't think about whether this fits or not fits. And that's the same, coming back to my example with safety. Are we really thinking about what we are comparing if you have a randomized trial where two different treatment durations are there, or in one trial people are dying earlier, so less efficacy, and so they have less time to get certain adverse events because they're just dying. And then you are claiming, oh, it's a more safe drug. Less, I do not know, less diarrhea or thrombocytopenia, but it's just because they have a shorter treatment. They didn't have the chance to get the disease. So at least, as I said, it's making it transparent, and also is the right way. Let's start the medics and the safety person or the pro colleagues first think about what is their question, and then we look for an answer.
There's a question here around missing data, Armin, which arguably is more to do with the how, our statistical analysis and our estimation, than it is around the what and the target of estimation. But nevertheless, the question's interesting because it differentiates between missing data that are unavoidable. If a patient, for example, moves house away from a study site and can't participate in the study any longer. And I guess the alternative to that is missing data that arise because of a discontinuation of treatment and a discontinuation from the study because of a progression of the disease or an adverse effect of the drug or whatever else it might be. Could you comment around different types of missing data, but from a sort of an Esterman and intercurrent event perspective, and perhaps repeat the differentiation between intercurrent events and missing data?
At the end, also an intercurrent event can lead to missing data. Let me phrase it differently. If you have an intercurrent event, take our progression-free, disease-free survival. And somebody gets a new anti-cancer therapy, and you haven't observed a relapse before. Then at the end, your exact measurement is missing because you wanted, if your research question is, I want to have, I do not want to be the treatment as an hypothetical estimate. I don't want that the estimation is affected by follow-up therapy. You have then now a problem because there is an intercurrent event. It disturbs your estimation of the disease-free survival interval. And what you to do, if you then ignore assessments after the intercurrent event, this intercurrent event leads to a missing information. And you also there have to decide what is the best estimation of this missing value. The example I gave you there was the simple approach. We censor the information. That means you assume that those patients who get intercurrent event would behave similar. You have an intercurrent event, that means follow-up therapy, would behave similar than those who didn't get follow-up therapy. Per se, a very doubtful assumption because a physician, by intention or by whatever reasons, gave the patient follow-up therapy before PD. This is not by chance. Maybe the patient is just before relapse, and I don't want to wait 8 more weeks to the next scan, gives the patient the drug. So that is an intercurrent event which disturbs, which leads to a missing information. So at the end, from a pure technical part, when you see the data, you don't know why your information is missing. So whether it's missing because the patient lost to follow up, or whether the patient is the information is missing because of an intercurrent event. However, you can use these two missings differently because if somebody lost to follow up, or you can there assume this is by random because the patient moved to a different location, compared, as I said, to an intercurrent event. So it's the how to phrase it, good result is more or less the same when you look at the data, but the reason for the intercurrent, for the missing is different. The one you can avoid, you can try to avoid. The other is part of the treatment. And it can give you a different information. Because when somebody gets therapy, it's rather likely that the patient is getting worse. A new therapy, compare that the patient is just lost to follow up because he was assigned to chemotherapy in a randomized trial. It's an open trial, and the patient then said, "Oh, I don't want to have chemotherapy." Is that a little bit answering your question?
Yeah, no, thanks for that, Armin. I think what it does do is show that the framework and the language that's used in the ICH E9R1, the addendum to E9, it's necessarily been written in a general way that to try and work across all therapeutic areas, all type of interventions, etc. And the language is a little bit new, a little bit technical. We have intercurrent events, and we have strategies, and we have attributes, and we have Estermans, and it will take an investment of time for people to become familiar with the words and the topics. Now, the Esterman itself is a description of the treatment effect that I want to estimate. And we deal with, or we specify that research question in respect of intercurrent events such as discontinuation of treatment or treatment switching that can happen. And those events can happen in clinical practice, they can happen in clinical trials. None of that requires the concept of missing data. The Esterman defining my research question, defining my target of estimation tells me what I would like to estimate. If I can't collect data that are relevant to that, then I have a missing data problem to address in my statistical analysis. But for example, when I'm talking about a research question, I might talk about treatment discontinuation, whereas study discontinuation is something different. That's a missing data problem. It's extremely important problem when it comes to estimation, but it's not part of how I define an estimand. Now, when I specify these intercurrent events, I can do that in a granular way. And I might want to talk about, for example, the reason for treatment discontinuation. I might want to talk about treatment discontinuations that are due to progression of disease, treatment discontinuations that are due to toxicities, treatment discontinuations that are due to the patient getting COVID-19 or the coronavirus pandemic more generally. I might want to specify these intercurrent events with more granularity, so that I can specify the research question more precisely, and perhaps deal with this or address this issue of the difference between a patient who stops treatment because they move away and they're out of the scope of the particular research center, from the patient who has a toxicity and needs to discontinue from the treatment. So it's an important distinction between intercurrent events and missing data. Okay, there's another general question here, Armin, around how do we measure success of the Esterman framework. So you have to get out your crystal ball and think about where we might like to get to, say in 5 years' time, and how would you measure? What would you look to judge whether the guideline and the framework has been helpful, has been a success?
That's a tricky question. The first answer would be, if nobody talks any longer, if it's in your DNA, so there are no further trainings on estimates because everybody knows it. And you would start in the journals a clear, let me say, structure following this. At the moment, also submission documents or journals are very endpoint focused. And if we would follow also in the reporting of the trials, the structure of the estimates framework, making clear what is the question I have answered. and not, as I said, when you at the moment look in journals, the summary, the primary endpoint was overall survival. The hazard ratio was, and that's the summary. So if we have this in the reporting, we are, let me say, that would be a success.
Thanks. And related to that, of course, there might be seen a risk that this is, this ends up as a rather sort of academic topic for statisticians to get excited about. And those of us who were responsible for initiating this can be blamed, can be criticized, and I hold my hand up for doing this under ICHE 9, under statistical principles for clinical trials, where your lecture and our discussion nicely illustrates that this is about so much more than statistics. This is the very fundamentals of why we're conducting clinical trials and defining treatment effects that we would like to estimate in those clinical trials. So how do we engage the broader community? How do we engage clinicians, academic researchers, and how do we bring those disciplines? How best do we bring other disciplines into the conversation and up to speed so that we can achieve our objectives and get where we'd like to be, having clarity on our trial objectives and alignment between those objectives and our analyses?
The key for success is to make clear what are the benefits of it. So what do I have? What do I get out of this? Except that I have to write a little bit more in the protocol and follow some structure, as Rob said earlier, a tick box. And this is at the end a little bit difficult to measure, but it helps. If it helps you to reflect a little bit on what you are doing and then define the research question in this way. For example, when we had this disease-free survival example, if I'm interested into the time until progression, irrespectively of further follow-up therapy, then I have to sample this information. Or a totally different question is for example, take an example of dose-finding trials in oncology. I have seen protocols where more or less it's written, we are counting dose-limiting toxicities. And is this really what we want to get, or is it more to get the dose-toxicity relationship? And if you want to get the dose-toxicity relationship, what do you have to measure and to get to achieve this objectives, so that you think a little bit more on your trial? Is the trial fit for this question compared to those limiting toxicities? Or an example there, an intercurrent event, for example, is the patient is taking off drug because the investigator is afraid that the patient might get a severe RA, which is a dose-limiting toxicity. That you reflect upfront, is it DLT or not a DLT, and then define your tolerable dose, thinking through all these events before and not do it by chance, by, let me say, what you observe. So you, in fact, I'm struggling a little bit. What is the benefit? The benefit is at the end that you, or take then that you really think upfront, what do I expect from the trial and what do I want to get out of the trial? Or coming back to this overall survival example, if you, to make clear, even if it's not new, what does it mean if I allow to switch the treatment? Or what does it mean if there's a change of landscape and I might see follow-up treatment which I want to avoid? At the moment, often this is done, there's a new drug which is in the approval process of the FDA. Let's go, it will be approved in May. Let's not go to US. Let's go to some different countries to avoid this follow-up treatment. this is harming your trial because you have then a different population. You implicitly avoid something by changing your population or make a different selection of countries. Rather say, okay, this drug will be approved in US. Let's go to US. This is my target population. But then I account in the analysis for this new drug and don't take a treatment policy estimate. I take a hypothetical estimate. So it's more thought through, and then you might be more successful because you made the trial in the population you are interested in.
I think that's a really important point, Armin, and it's one that was a driver for me as we were developing this, that perhaps in a misguided way, trials have reduced their external validity by trying to avoid these intercurrent events, these events that do occur in practice. And the framework and the way that we can now discuss research questions and statistical analyses and data collection that are aligned to those may help us address concerns over external validity of trials. I've got a couple more questions in the Q&A. One is about the whether there's a requirement to apply the Esterman framework to ongoing protocols, and whether journals, and perhaps we can extend that to regulatory agencies and HTA bodies as well, will require future submissions, so of a trial for publication or of an application for a marketing authorization, to incorporate Esterman's language, even though the trials were designed prior to the introduction of the addendum.
I mean, it's never too late. So I would say it's good to apply it even in running trials. It's never too late to reflect on what you are doing. So, and the so if, for example, you were designing your overall survival trial and thought, okay, I'm fine with using all follow-up treatment, and then you see it's an example from the past, was a lung cancer trial, and then TKIs were introduced in Asia. So in Asian patients, now TKIs were available, and then we analyzed the trial, and all patients in the control arm got TKIs and nobody in the experimental arm. So, oops. At that time, that was pre-Esterman's time, what do we do? Here, the Esterman framework would give some thoughts and argumentation support to what was my initial question and how should I answer this? Or take anti-drug antibodies. They occur, but only in one arm. So to compare to what? What is my, and there the Esterman framework would help you to define a kind of similar population in the control arm by defining those patients who under the treatment would have an anti-drug ADA to have a comparable comparator. It's a helpful tool to think this through. So, and it's never too late to think things through.
No, I think that's right, Armin. Sorry to interrupt, but I think that's right. As a sponsor, I would reflect on my open and ongoing protocols and make sure I was doing sensible things, answering the relevant questions with methods that were aligned. But my suspicion is that there'll be quite some flexibility from regulators, HTAs, and journals if companies haven't used the framework because trials were already ongoing before the addendum was finalized. Still, you know, once you've turned your brain to this way of thinking, it's hard to switch it off. And as you say, light may be shone on particular problems that are now hard to ignore, whether the trial has used the the framework or not. And the sort of poster child example for me is imputation or prediction of values for a particular variable after a patient has died. This is relatively common in clinical trial analysis, that even for patients who've died, because we want to adhere to what we think is the relevant ITT principle, we make predictions or make imputations for measures on a particular variable after the patient has died, and that doesn't seem particularly smart. It doesn't seem to align to a particularly relevant research question. Okay. There's a group of questions. There's a group of questions now around the perspectives from different stakeholders. So one relates. The thrust of one question is essentially, shouldn't all stakeholders align on the research question of interest, aligning to the question that really reflects a true unmet medical need in a particular context? Another question comes from a slightly different perspective and says, well, if different stakeholders do have different research questions and hence different estimands, is it okay to have one as primary and another as secondary or supplementary? And a third related question, perhaps looking more to the future, says, would you expect key estimands in the oncology area to align after a while, given there's a rather sort of standard set of existing endpoints and analyses that are in use?
Let me phrase it. One does not fit all. So coming to, I'm personally totally fine if different groups have different questions. Because the research question of a patient might be different than of a clinician. And so I'm in principle totally fine if you have multiple research questions. It's not about to get everybody in one basket and have the one and only question. It's rather, and we have this already now, that when you go for HTA bodies, you use a different analysis than when you go to the regulator. So I would say phrase the research question for the patient, for let me say, the person who is interested in. And this includes Esterman framework, also the measurement, the population level summary. For example, I don't want to go into crossing curves, but in principle, for example, a hazard ratio is a good population level summary, but maybe a patient is more interested on, I don't really want to have a risk reduction. It's more of the question, do I live in 5 years? So it's totally fine to have different questions for different stakeholders, as long as they are not contradicting and harming, or could be answered in the trial itself. The special, when it comes to key estimates, compared to other indications in oncology, we have at the end a very narrow world. We have somehow an early endpoint, tumor shrinkage, often measured via response rate. We have time until the tumor grows again after treatment, PFS, disease-free survival, something like that. And we have overall survival. Having said that, we have anyhow at the end 3 major types of endpoints. And if you then look what you can modulate in the estimates framework, maybe per endpoint, 3 or 4 research questions. So it's a limited number anyhow. So that's why I would say we will come to a kind of standard, which also increase comparability. And
sorry, Armin, please finish.
It may be one of the critics in oncology of estimates in oncology that we up to now not really found something new. So at the moment we are still in the status of, let me say, squeezing the estimates framework into the current landscape, but we haven't thought about out of the box, and me too, to find something totally new. So
I think that's a really interesting observation, because as you say, there already are different research questions that are posed around even OS data for different stakeholders. The FDA and the EMA guidelines around analysis of PFS essentially describe slightly different research questions. I think in terms of quality of life and PRO data, my experience from the time in the regulatory system is that they got nowhere near enough attention in the relevant research question to phrase, and then how that would translate into how we use the data and the statistical analyses we do. And there's huge room for improvement with that type of data. And perhaps the fourth category that's interesting is quantifying treatment effects on duration of response. So anytime we look at objective response rates or remission rates in hematology, we might also be interested in the durability of that effect and the duration of response. And actually calculating a treatment effect, even from a randomized trial, calculating a treatment effect on duration of response is not done, or is not done well. And Armin essentially is a principal stratum question, and we have different responders in one arm than we do in another arm, and a naive comparison between response durations in one set of responders to response durations in a set of responders on a control arm would not describe a treatment effect. So I think it's true that the world of oncology, hematology hasn't been revolutionized yet, but I think there's a long way to go in some aspects of this work.
And especially duration of response is a tricky one, because think about, you have two drugs, and one has and we are late line patients, no effect at all. We have one drug which gives 20% of the patients 8 years survival or duration of response, and another drug is 40% of the patients 4 years. So the average benefit of a patient is the same, but it's totally different. How can you compare them? And that's what is better? And there, coming back to this initial question, different people might have a different focus on what is an effect and how you measure it.
There's actually a related question appeared in the Q&A, whether we can comment on the appropriateness of principal stratum Esterman. So Estermans that invoke the principal stratum strategy based on early tumor responses or other post-baseline biomarkers. Would we ever be interested in treatment effects on downstream variables such as progression times or survival times in a principal stratum of patients who respond or have a particular change in a biomarker?
In principle, that's a potential way, or we should go in this. Coming back to the example, you have maybe chemotherapy, or radiation. It's working in every patient, and you have an average effect, and you have a biomarker driven drug where you have at the end no effect in 70% of the patients and a super effect in 30% of the patients. So now the principal stratum would give you the question, or the counter question would to find the same population in your control arm which are responders in your new drug, because this you want to compare at the end, or who have a certain, especially post-baseline assessment. So the principal stratum approach helps you a little bit to compare a stratum in your experimental arm to a similar stratum in the control arm. The tricky thing is to identify the stratum in the control arm, because it's normally not a baseline characteristics. For example, you see in patients, certain patients, and you don't know why, that they have a higher exposure. Or coming to Erbitux is a drug which causes skin. You have rash, and people with rash live longer. But it's not the rash why the people are living longer, and you have no clue at the why is you up front you don't know whether a patient will get rash or not. It's an indicator. It's a stratum. It's but what is the complement? What is when you make a randomized trial? So sorry for the not straightforward answer. It's the right way to go. At the moment, I haven't a solution.
I think often these principal stratum will be secondary questions, but that doesn't preclude their usefulness to gain insights into treatments. And as you say, if we have a trial comparing a PD-L1 inhibitor that has a different profile of objective responses and duration of responses against a chemotherapy regimen, then if we want to know, if we want to try and estimate effects on duration of responses, we have to use this type of approach. Otherwise, we're not making a relevant research question. We're not generating a causal treatment effect. We're not defining a causal treatment effect. And similarly, it may be of interest to say, actually, if a patient does get a rash, are they the ones who seem to go on and do better? But if we want to answer that question, we have to compare apples with apples. We can't compare the patients who get rash on one treatment with the patients who get rash on another treatment. That doesn't necessarily compare apples with apples and define a causal treatment effect. So I think the point here is that principal stratum questions can be of interest. And if we want to define them, we now have a language for doing that. And we now have we're starting to build a set of tools and statistical analyses that help us generate unbiased estimates and avoid naive comparisons that don't define causal treatment effects or give unbiased estimates of effect. Now, Axel, I see you've appeared, which is a pleasure, of course, but I fear it might be bad news in respect of timing.
Yes, it is in a way. I mean, maybe we can go through the last questions with short answers that all our participants get an answer, but we don't go too much over the end of the meeting. In Europe, it's pretty late and people might wish to join their families.
That's understood. And I've resisted saying hello to a number of old friends who've appeared in the Q&A, and I'll keep resisting that as I ask a question. Just coming back to this question, Armin, of different stakeholders having different research questions, different priorities for Estermans. Do you think that the Esterman framework has the potential to offer an opportunity to reduce these differences, bearing in mind that what we all really want to know is the magnitude of the treatment effect when it's applied in real life, in clinical practice?
I'm not so convinced. I'm not so convinced because why didn't we manage to do this up to now? And think about the example I gave with the 3 trials on disease-free survival. Is there and how to deal with the intercurrent event? I don't think it will be natural. For example, when you had this second primary malignancy, to use or not to use. I don't think in the endpoint definitions, I'm not so optimistic. I should make it short. I don't think so. Because we make differences more transparent with the Esterman, so I'm not so convinced.
Time will tell.
Time will
tell.
I would be happy, but I'm not convinced.
And perhaps the final question from the Q&A, Axel, that we pose is, Just for final reflections from your side, Armin, on the consequences of the framework in terms of benefit risk discussions and the role of Esterman's and sort of more precisely defined research questions in benefit risk discussions. So beyond just results of primary and secondary analyses.
It's all about benefit risk, that's for clear. And the logical consequence is if you want to apply estimates for benefit risk, we also have to apply it for safety. And this is the, that I would say it's the next step. Let's apply it for safety, and then we can come to benefit risk.
Because,
sorry.
So sorry, Armin. Please.
No. Go ahead.
Apologies. I thought you were stopping. No, and I think that is a sort of nice way to finish, because what we're talking about is understanding treatment effects, defining treatment effects that are interesting to learn about, and then defining them in a way that we can align our statistical analyses and actually give a good estimate of them. And nobody says those treatment effects have to be positive, so have to represent benefit. And nobody says those treatment effects have to be coming from RCTs. This discussion is relevant to toxicities where we aim to estimate a treatment effect, define and estimate a treatment effect. And so can be relevant to, or is relevant to the way that we understand benefits and risks and hence make the trade-offs between them and the stakeholders make their decisions. And it's relevant to effects that will be estimated based on data generated in single-arm trials, or as per the question earlier, in meta-analyses. If we're defining a treatment effect, it's above and beyond consideration of the experiment that will be used to estimate it. And I think that's a sort of elucidation of why the framework is powerful, because we get that clarity on what we're trying to do before we're tied down a little bit by any operational considerations and historical precedent about how we do it. So it gives us the opportunity to open our minds and perhaps do something a little bit different. So final comment from my side, again, this is not doing what we've always done, just using different words to describe it. This is an opportunity to reflect on what it is we try to learn about medicinal products, about our experimental treatments, and for the public sector stakeholders, the regulators, the HTAs, and then down to the payers, the prescribers, the patients, etc., to reflect on what it really is we want to know about treatments, and hopefully this Esterman framework and language gives us a good basis to have those discussions. And Axel, thanks again to you and the CDDF team for giving space to excuse me. Thanks to you and the CDDF team for giving space to this important topic, and thanks to Armin for the informative lecture.
Yeah, thank you very much, Rob and Armin. I think that has been an outstanding evening, and very, very detailed, very good information. I hope that will be very valuable to all our attendees. So many thanks to you. The slides will be available on the websites together with the video for CDDF members. It will be open to everyone after 12 months. And we would like to flash our upcoming webinar, which is about patient access to newly registered anti-cancer agents on the 9th of December, next week. So we're looking forward to see you again there. And many, many thanks to Rob and Armin for this fantastic presentation and discussion. Have a good evening, everyone. Bye-bye.